All right, let's try 8. So it says, based on the information presented, which of the following is the most likely explanation for a buildup of cholesterol in the blood of an animal? So remember, our bodies are like this, where we have blood, including blood cells, in like a tube. And on the side of each blood are these different cells. So if there is a buildup of cholesterol, that means cholesterol is stuck in the blood and not going into the cells. So that's, without even reading the background information, I can figure out this question. It says the animal's body cells are defective, so not working, in exocytosis. Well, no, that means leaving the cell and going into the blood, so it's not A. B, the animal's body cells are defective, not working, in endocytosis. So like I said, this process is not working. Endocytosis means into the cell, so it's likely B. Let's just check. The animal's body cells are defective in cholesterol synthesis. No, because we still have buildup of it in the blood. The animal's body cells are defective in phospholipid synthesis. That's the cell membrane, what it's made out of. And that has nothing to do with building up cholesterol, so it is B. All right, number 9. Which of the following scientific questions is most relevant to the model represented in the figure above? First, let's figure out what the heck is going on here. So here we have these different substances that are moving across the membrane in different ways. So let's figure out those ways. Here, this is simple diffusion, because we see this molecule is going straight through the phospholipid bilayer down its concentration gradient with no protein. So this has to be a small nonpolar molecule, or water, which is polar but tiny, so it can squeeze past the nonpolar tails. Okay, here we have facilitated diffusion. And these are both types of passive transport. Facilitated diffusion, it's still going down its concentration gradient from high to low, but these are either too big or polar or charged, so they can't go straight through the phospholipid bilayer. They need a protein. And last but not least, we have going against the concentration gradient from low to high concentration, so this is active transport. So now, I'm trying to figure out what questions can be answered by this. Is ATP required for transportation of sugars across the outer mitochondrial membrane? Well, actually, let's look at this. This says extracellular fluid. Extracellular means outside of cell. This says cytosol. That's the same as the cytoplasm, so this is inside the cell. This is not the mitochondrial membrane, so it's not A. B, do the types of phospholipids in a membrane affect the rate at which molecules enter a cell by passive diffusion? Well, it's not actually showing me different types of these phospholipids, so no. C, which molecular substance is actively transported across the plasma membrane? Well, we know that this one is actively transported, so yes, that does look like something we can answer with this. And then D says, how does temperature affect the movement of molecules into lysosomes? So this is, that's wrong, and it doesn't talk about temperature, so it's not D, it is C. Number 10, this one has to do probably with like osmosis or cell transport, just based on this picture where we have water and then water with different percentages of different solutes. So it says the following questions refer to an experiment in which a dialysis tubing bag, so this is just a bag that is semi-permeable, with a mixture of 3% starch and 3% glucose, is placed in a beaker of distilled water. So remember, this one has solute, this one no solute. After 3 hours, glucose can be detected in the water. So glucose can go through this permeable membrane outside the dialysis tubing bag, but starch cannot. So starch can't go through, glucose can. From the initial conditions and results described, which of the following is a logical conclusion? So A, the initial concentration of glucose in the bag is higher than the initial concentration of starch in the bag. Well, no, they were both 3%, so no. B, the pores of the bag are larger than the glucose molecules. That makes sense because glucose could leave, but smaller than the starch molecules. That actually makes sense because the starch could not leave. So pores are holes that things can go through. So starch could not go through those because they were too small for the starch molecules. C, the bag is not selectively permeable. Remember, we use that to describe membranes. It means some things can go through it and other things cannot. This one, it says glucose can go through it, but starch cannot. That is the definition of selectively permeable, so it is not C. D, a net movement of water into the beaker has occurred. So if I have my setup like this, and water is going, it's saying going into the beaker, well, the beaker has to have higher solute concentration, but that's not the case. In this, it says that this has higher solute concentration, so water should be moving into it. So no, not D. And then E says the molarity of the solution in the bag and the molarity of the solution in the surrounding beaker are the same. Well, molarity means how much solute is in liters of water, mole per liter. And we know that this had 3% starch and glucose, and this has 0% starch and glucose. So no, it is B. Number 11. Which of the following best describes the condition expected after 24 hours? So we expect it to kind of reach equilibrium at this point. It says, which of the A, the bag will contain more water than it did in the original condition. Well, that makes sense. Water follows solute. There's more solute there, so that one's looking good right off the bat. Let's check. B, the contents of the bag will have the same osmotic concentration, so the same amount of solute and water potential as the surrounding solution. Well, we know starch can't leave here, and this doesn't have any starch, so that's not going to be able to balance out, so it's not going to have the same osmotic concentration. Even though we're trying to reach equilibrium, we actually can't because the starch is stuck in here and can't go out here. C, water potential in the bag will be greater than the water potential in the solution. Well, higher water potential means it's more likely to move. We know that water is going to go in, not go out, so no. D, starch molecules will continue to pass through the bag. That could never happen. It said that in the background, so no. And then E, a glucose test on the solution in the bag will be negative. Well, no, because glucose, even though it is leaving, is going to then reach its equilibrium where it's going in and out equally. So it's not going to be negative. There's still going to be some glucose in there. So it is, in fact, A. Number 12, if instead of the bag, a potato slice were put into a beaker of a distilled water. So that's a potato. Potatoes have starch, so we know there's starch in there. Right? It's a starchy root vegetable. Which of the following would be true of the potato slice? Well, if this has more solute and it's placed in distilled water, water is going to go in, so will it gain mass? Yes. A is most likely correct. Let's check the others. It will neither gain nor lose mass. No, because water is going in. It would absorb solutes from the surrounding liquid. There's no solutes in distilled water. It would lose water until water potential inside the cell equals 0. Inside the cells is equal to 0. So it's saying that water is going to continue to go out of it. Out, 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 out. until water potential is 0, but we know water is going to be moving into it, so no. The cells of the potato would increase their metabolic activity. This has nothing to do with metabolic activity. This is about osmosis, so it is A.